Okay. Hello, everyone. Thank you for coming to the graduate seminar series today. Uh, as you know, we've moved it to a virtual platform. I think just for ease of everyone who may be working remotely or for any other reasons, but we'll be recording these so anyone who is unable to join can watch afterwards. So I think that's a nice perk of doing this on Zoom. Uh, my name is Brendan, and I am a part of the Agronomy Graduate Student Organization. Um, so if you have any questions about these presentations moving forward, go ahead and let me know. Um, and we're going to use the same Zoom link every Friday for each of the presentations at 10 a.m. on Fridays. And then for today, if you have any questions for the presenter, feel free to write them in the chat or save them for afterwards, and you can express them verbally. Yeah, and then on that same note, please just remember to keep yourself muted so you're not interrupting. Yeah, and so today we have Frank Johnson, who is a part of Dr. Shalimar Armstrong's lab. He received his PhD last December, and he will be speaking to us today about some denitrification of agricultural soils, as you can see from his presentation here that he's sharing. And so without further ado, I will let him get started.
Well, thank you so much for that introduction. And I'm really excited to be speaking with you today. Today, my presentation is titled Investigating Nitrification from Two Missouri Claypan Soils. And I will be discussing a part of my dissertation research that I conducted at the University of Missouri in Columbia, Missouri. Here's an outline of how the presentation is formatted today. First, I'll start with an introduction, followed by objectives and our hypotheses. Then I'll discuss where we did our research, where we conducted our research, followed by results and discussion. And last but not least, the conclusions. Here's an illustration of the nitrogen. As many of you know, nitrogen has many fates once it enters a soil system or an agricultural system. It's usually applied as fertilizer in the form of nitrate or ammonia. Nitrate can then be immobilized. Went back. There we go. Nitrate can then be immobilized into soil organic matter, which can then undergo mineralization and release ammonium. Ammonium can be lost through volatilization or get fixed between clay particles. And then ammonium can also undergo nitrification, in which ammonium is converted to nitrate. And then nitrate can be transformed to nitrous oxide and dinitrogen through denitrification. And that is the portion of the nitrogen cycle that I am particularly interested in. So denitrification is the microbial facilitated reduction of nitrate to dinitrogen. As you can see, it's along this pathway here in the green box, starting as nitrate and ending up as N2. There are intermediate gases that are produced during this process. So we have nitrogen dioxide, then it is transformed to nitric oxide, followed by nitrous oxide before the last transformation to dinitrogen. Now, nitrous oxide is an important greenhouse gas because it is an important gas in this process because it is a greenhouse gas with a global warming potential of about 300 times that of carbon dioxide. And it is also an ozone depleting gas. So it reacts with ozone in the atmosphere. Not only that, but it stays in the atmosphere for about 15 to 18 years before it starts breaking down. And so it stays in the atmosphere a little bit longer than some of the other greenhouse gases. Here on the right side are genes that code for these enzymes here on the left side. That are responsible for facilitating the reaction. I have them listed here because it was another portion of my dissertation research that I won't necessarily be discussing today. But if anybody has any questions about the molecular work that I've done, please feel free to ask as well. So what influences denitrification? First and foremost is soil water content. A water-filled pore space of at least 60% is required for denitrification to start. And it typically occurs in saturated and anoxic soils. Typically, the longer the soils are saturated, the more denitrification it will occur and is likely to occur. Next, we have soil temperature. Denitrification typically occurs at warmer temperatures. There was a study conducted where they increased temperature by 3 degrees Celsius, and the amount of denitrification that occurred nearly doubled. Soil pH, which of course impacts a lot of different areas in agricultural systems, definitely impacts denitrification. It typically occurs between a pH of 6 and 8. However, there have been instances where denitrification occurred at as low as a pH of 4 or 5, and that was most likely due to the denitrifying organisms adapting to the acidic conditions. Soil organic carbon, the labile carbon content of the soil, as you all know, that impacts any part of or any microbial activity in systems. And last but not least, we have the soil nitrate concentration. Nitrate is the starting substrate of the denitrification process. So the more or the higher the concentration is of soil nitrate, the more opportunities there are for it to be transformed into denitrify. As I mentioned in the previous slides, there are intermediate gases that are produced along the denitrification pathway, or it can go all the way to the end, ultimately ending up as dinitrogen. So it led us to believe, and we question, what would cause microorganisms to transform this nitrate all stop at N2O. And so we know that soil water content, soil temperature, and soil pH were all factors that influence denitrification. And the answer that we came to was energy. So we decided to take some time to look at the energetics of denitrifying microorganisms. There was a study conducted by Koike and Hattori in 1975, and they observed that the number of ATP that are produced when these transformations occur are associated with the same oxidation state of nitrogen in this particular process. So when nitrate is converted to nitrogen dioxide, the number of ATP that are produced are 5. And then when nitrous oxide is converted to dinitrogen, only 1 ATP is produced. So that means that these microorganisms would only complete denitrification and transform nitrous oxide to dinitrogen when it was absolutely necessary, because it wasn't energy efficient for them. It was more energy efficient for them to convert nitrate to nitrogen dioxide. There is a huge need for this research because corn is grown on approximately 1.2 million hectares in Missouri and 36 million hectares in the United States. Corn is nitrogen to grow well, so as a result, that's why it receives the highest amount of nitrogen fertilizer in the US. The NRCS estimated that between 5 and 20% of the applied nitrogen fertilizer is lost through denitrification. A little bit more about agriculture and greenhouse gases. The agricultural sector accounts for approximately 7% of the total US greenhouse gas emissions. That's compared to like the electric power industry, which contributes about 34.6. Although it accounts for only 7% of the total greenhouse gas emissions, it's responsible for 79% of the nitrous oxide emissions and 35% of the methane emissions. And a lot of the nitrous oxide emissions can be attributed to soil management practices, and those account for 92% of the total US agricultural nitrous oxide emissions. Those management practices includes the fertilizer use and type, whether using NOR, and the tillage practices that are conducted. So those management practices can have the opportunity to increase or decrease nitrous oxide emissions from agricultural systems. The merit for my research that I'm discussing today is this study conducted by Blevins et al. In 1996. And so what they did is they used N15, or an isotope of N that they could track throughout the growing season. And at the end of each growing season, they tried to account for all the nitrogen that they applied in the two fields. And at the end of the 2 growing seasons, they observed that 30% of the nitrogen was in the saturated zone of the soil, 27.3% was in the grain, 5% and 5% was in the unsaturated zone. That left roughly 38% of the applied nitrogen unaccounted for. Now, they did not take gas measurements or measure nitrous oxide. And so they believed that a majority, if not all, well, all of it was lost through denitrification. There were three objectives to my dissertation research. First is to use the denitrification enzyme activity for measuring the spatial variation of denitrification potential in two different Missouri fields and establish a relationship to actual denitrification rates. The second one was to relate abundance of Nazi gene measurements by 3 RNA sequencing methods to the denitrification emissions occurring from the fields from our soil cores. And then my third objective was to upscale actual denitrification flux data to make field scale estimates of annual denitrification and determine its importance to the end budget. So the only objective that I'm discussing today is this first one, using the denitrification enzyme assay. So our hypothesis was that denitrification is a major loss pathway of fertilizer in our overarching hypothesis. In that Blood et al. Study, they did not account for 38% of the nitrogen that they applied in their field. So, and they assumed that most of it was lost through denitrification. So then we thought, and the question that we were looking to answer was, to what extent does topography control the temporal and spatial occurrence of denitrification? And we believed that landscape position influences soil properties that control denitrification. And those soil properties include how the nutrients move by landscape position. That includes the organic carbon deposits, which is, has implications about the microbial community, and even influences the way water moves through these systems. So next, I'll discuss where we conducted our research and our site information. But first, I have to discuss LTAR, which stands for the Long-Term Agroecosystem Research. It is a network that consists of 60 colleges and universities, several federal agencies and state agencies, industry organizations and international organizations, all with the mission of being enabling to understanding and forecasting of regional landscape capacities to provide agricultural commodities and ecosystem services under changing conditions. As you can see from this map, there are sites all over the United States. There are some in the Northeast and in the Northwest, and even quite a few in the South. Our particular site was located here at the Central Mississippi River Basin. So there was a lot of research already being conducted for the LTAR network here. And so we asked our question, I asked myself a question: how can my dissertation research contribute to the larger project that was going on here? And so here at this site, we have two fields. We've dubbed them Field 1 and Field 3. Field 1 is our aspirational system under no-till, a corn, soybean, wheat rotation, and a variable nitrogen rate application. And our Field 3 is what we've dubbed as our business as usual system, which is under a disc tillage and a corn, soybean, soybean rotation. And both of these fields, we have Claypan soils with a Mexico Leonard Armstrong Association. I just want to explain what Claypan soils are, just in case nobody knows what they are. But whenever you, or if somebody has a question about what they are, there we go. So a Claypan soil is technically characterized as, once you get down to a soil horizon and the profile where the clay content is 100% greater than the horizon directly above it. So here we have a clay-less parent material with smectitic mineralogy. Smectitic clays are 2 to 1, and they are shrink swell clays. So as it rains, the soil swell, and as they dry, they shrink, and they can result in cracking within the fields. These fields are typically poorly drained. Claypan soils are typically poorly drained. And here are our common soil series were Putnam or AGSO silt loam in the summit, Mexico silt loam in the shoulder, Leonard silt loam in our side slopes, and Montauk silt loam in our toe slopes. Here is an aerial photo of our Field 1, our aspirational system under the corn, soybean, wheat rotation. And so typically our summits were here on the edge of the field, and you would go down the landscape position as you would get to this drainageway here through the middle. Here's our business as usual with the corn, soybean, soybean rotation with our toe slopes resulting down here in the side of the field. So on these two fields, I conducted the nitrification enzyme activity assay on 90-meter grid samples. In field 1, we ended up with about 48 samples, and field 3 with 44. Now, there were samples that were taken at the edge of field 1 to the west that are not shown in this illustration. They were taken about over here. And as you can see there, we've delineated by landscape position here. So we have our summit, our back slope, and our toe slope. Now our shoulder positions are included in the back slope, and we did that because they are both areas of loss in terms of losing nutrients down the landscape position. And our landscape delineation was objectively developed using light detection ranging data, or LiDAR data, plus other characteristics that are known about these two fields since we've been working on them for so long. And on these 90-meter grid samples, I performed the nitrification enzyme activity assay, which measures the nitrification potential or nitrous oxide production under conditions in which temperature, nitrate, and carbon are not limiting factors. In this case, our carbon source is glucose, our nitrogen source was potassium nitrate, and I also added chloramphenicol, which is an antibiotic, and that was added to halt microbial reproduction. Initial soil characteristics were also obtained from these samples. The samples were sent to the soil plant and testing lab on the University of Missouri's campus. I mentioned earlier that my project was a part of a larger project at this LTAR site. And so this is what they were doing. And so they have 2 landscape transects per field in which they were taking actual denitrification measurements from. And here they are in field 1 and field 3. There were 6 cores per transect collected in triplicates for a total of 36 cores. 12 of the cores were shipped to Dr. Peter Groffman and his team in New York for direct denitrification measurements using a gas flow soil core incubation system. They were using the N-Farm, which is the nitrogen-free atmospheric recirculation method. And the gases that they measured were nitrous oxide and dinitrogen. And they measured them at varying O2 levels at 0% oxygen and at 20% oxygen. All statistical analysis conducted on this data was done using SAS and the necessary procedures to determine normality and conduct analysis variance and the different mean comparison test. Now here's to the fun part, the results. So first are the denitrification potential results. Here on our x-axis, we have field, and our y-axis, we have our nitrous oxide flux in kilograms of nitrogen per hectare per day. So as you can see that there were no significant differences between the two fields. They were both just above 1.0 kilograms of nitrogen per hectare per day. However, field 3 had higher variability in denitrification potential compared to field 1. So we took it a step further and looked at landscape position, how landscape position impacted denitrification potential in each of these fields. So on our x-axis here, we have landscape position for our summit, back slope, and toe slope. And on our y-axis, we have nitrous oxide flux in kilograms of nitrogen per hectare per day. And in field 1, well, in both fields, denitrification potential was highest in the toe slope. But for field 1, the toe slope was statistically significantly higher than the denitrification potential in the summit position. And in field 3, the denitrification potential in the toe slope was statistically significantly higher than the denitrification potential in the back slope landscape position. Now, regarding actual denitrification measurements, here we have field in our x-axis in total denitrification, which is the amount of nitrous oxide plus dinitrogen on our y-axis. And it is also partitioned by both of those compounds. And for field 1, our total denitrification was about just under 0.2 kilograms of nitrogen per hectare per day. And for field 3, it was roughly 0.3 kilograms of nitrogen per hectare per day. But the most important thing to take from this figure is that in both fields, a majority of the gas that was produced was dinitrogen. And so N2 accounted for roughly 90% of the total denitrification that occurred in both of these fields. So we wanted to see if there was a similar trend between the denitrification potential and actual denitrification, the total denitrification that was measured. So here, the denitrification potential measurements are from the same samples that the N2O and N2, or the actual denitrification measurements, were measured from. So here on our x-axis, we have landscape position, and our y-axis, we have denitrification flux. I'm not quite sure what's going on with that formatting right there. But here in field 1, we see similar trends in denitrification potential and actual denitrification measurements. The lowest measurements were in the summit, and the highest for denitrification potential and actual denitrification were in the toe slope. Now for field 3, there was a similar trend for each of them, with denitrification potential being the highest in the toe slope along with actual denitrification, and then them both being measured lowest in the back slope landscape position. So that's something that we found very interesting and very compelling. So we observed that denitrification potential flux generally increased going down landscape position, and typically resulting, well, in both fields, were measured highest in the toe slope landscape position, and they were not different between the two fields. These results were opposite of Groffman et al. And Pareja-Sanchez et al. Groffman et al. Observed greater denitrification enzyme activity in the summit landscape position, but in their study, they were also looking at the burning impacts in a tall grass prairie, which is very different than our alpha saw Claypan system. Pareja-Sanchez observed that denitrification potential was impacted by tillage, application, timing, and the type of nitrogen fertilizer that was applied. Despite our contrasting management, fluxes were similar between the two fields. Regarding actual denitrification, it was also not different between the two fields, and was much lower than denitrification potential. But as I mentioned previously, greater than 90% of the total denitrification emissions were N2. This is opposite of Fiorini et al., McKenzie et al., and Liu et al., who all either observed an increase or decrease in nitrous oxide emissions when comparing no-till to conventional tillage. In both of our systems, actual nitrous oxide flux was low, and a majority of the emissions were N2. So it did not seem to impact N2O emissions. In conclusion, in both of our fields, denitrification potential was spatially dependent, and a majority of both fields had flux rates less than 1.5 kilograms of nitrogen per hectare per day. For actual denitrification, N2 was the dominant gas produced, and fluxes were 2 to 5 times lower than denitrification potential. Remember, N2 was predominantly produced. These are Claypan soils, and so they are poorly drained. So there were likely longer durations of saturation and ponding, which would have contributed to that complete transformation of nitrate to dinitrogen. And as a result, spatial patterns of denitrification in these fields reflect long-term erosion patterns in these two fields. Excuse me. However, more research should be conducted to better understand the impact management practices have on the spatial distribution of denitrification. Regarding my future research here at Purdue, there are 3 objectives, or 3 main areas that I'm interested in pursuing. First is understanding how different cover crop species influence actual and potential denitrification flux. The literature has been relatively split on these results. Some studies show an increase in denitrification and denitrification potential due to cover crops retaining a lot of the nitrogen in the system. So there are more opportunities for denitrification. And then other literature showed that there was a decrease. However, studies usually generalize cover crops instead of examining the different species. During my dissertation, one of the major components was using next generation sequencing techniques to measure NOS-Z gene abundance, which is the gene that codes for nitrous oxide reductase, responsible for converting N2O to N2. We were also interested in measuring NOR-B, which is the gene that codes for nitric oxide reductase, responsible for coding nitric oxide to nitrous oxide. However, we could not get our NOR-B primers to work. So being able to successfully measure and quantify NORB gene abundance NAZ along with the other genes along the denitrification pathway is important for us to be able to correlate functional denitrification gene quantification to actual denitrification emissions. So understand the molecular aspect. And then the last that I mentioned briefly was modeling denitrification. I developed an empirical model for the two fields in estimated denitrification over growing seasons when corn or wheat were grown. But there are several models out there, such as DSAT and DACENT, that have functionality to model denitrification. And if these models can account for the spatial variability and the spatial distribution of denitrification, they can potentially be useful when farmers are making management decisions in their fields. Now, I would like to thank everybody on this slide. Dr. Lesh and Dr. Motovali were my co-advisors during my dissertation. Dr. Vume and Dr. Scarf were also on my committee. Dr. Groffman and his team in New York, who helped analyze quite a few of the samples. I have to thank the USDA ARS, who funded my dissertation and this project. And then I also have to thank Dr. Shalimar Armstrong in the Sin Lab. I know there are some people from here that are joining us today for this presentation, and I'm looking forward to, and I've been enjoying working with you all. And last but not least, my family and friends who have supported me every step of the way. And without them, I wouldn't be where I am today. Oh, it ended. Okay, so with that, are there any questions? Here we go, I'm open for questions.
Thank you for that presentation. One thing I was wondering is why do you think there is a difference between the denitrification rates in the summit versus backslope between the two fields? Why that relationship isn't the same for both fields?
Yeah, that is a great question. I think it has to do probably with like the percentage of the slope. The summit landscape position is typically stable. So I did kind of expect, at least in one of the fields, for denitrification potential to be higher, just because it's not necessarily losing nearly as much carbon, nitrogen. Water is not necessarily moving away from it as much as it is in the back slope. So I think that is probably something that is worth investigating a little bit more that they should do. But then it could also be the depth to Claypan. If the depth to the Claypan layer is closer to the soil surface, then ponding is more likely to occur in that spot, which would result in higher denitrification. But I think that is a question that they should look further into in those two fields.
Hi, Frank. Great talk. I wasn't paying close attention when you were talking about the next generation sequencing stuff. So were you just doing DNA work to measure how much of the gene sequence was present in the soil, or was it actually RNA expression?
It was RNA expression, but so RNA was extracted and then analyzed with droplet digital polymerase chain reaction, nanostring sequencing, and RT-qPCR. And so basically what we were trying to do was correlate it. I didn't get too far, speak to too many details in this presentation, but basically we were trying to determine which of those sequencing techniques were best to measure these genes along the denitrification pathway. So we really wanted to relate them to other characteristics of our study. So we wanted to see if we observed the same spatial pattern. We wanted to see if they correlated to some of our initial soil characteristics that we observed. Unfortunately, that portion of my dissertation raised more questions than answered questions.
That could happen.
Yes, and so that is also why I'm interested in pursuing that area further here, especially because we were unable to successfully measure the quantities of NORB in our samples.
Right. Yeah, those things would be tricky unless you have a way to quantify how many bacteria are are there, you need to sort of know how much expression there is per cell to correlate those things, I think. But anyway, yeah, that's cool that you explored that. And yeah, nice talk.
See
All right, I raised my hand. And,
okay, there we go. Hey, I was trying to find you on the screen.
yeah, that's all right. I'm not gonna. I didn't want to talk over anybody. So unfortunately, I had another commitment and couldn't join until kind of the mid part of your presentation. So you may have covered it, but were there differences in among those landscape positions and fields in soluble organic carbon or nitrate that would help explain some of the differences in the actual denitrification that you measured, as well as maybe water holding capacity?
So I was looking at I prepared a correlation matrix, and they weren't statistically significant, but looking at the trends, they trended the same way. So yes, organic carbon and nitrate concentration were typically greater in our toe slope landscape position.
And, you know, the depth to the restricting layer is interesting. I mean, that could apply here to in depth to the till, et cetera. Were there other differences in the physical properties of the surface soils at these locations, or was that fairly consistent?
That was fairly consistent. I wish we would have taken some depth to Claypan data because I think that would have explained or helped explain a lot more. But yeah, no, the surface soils were relatively similar. They were contrasting management. I believe field 1 was under that aspirational system since I believe about 2015, 2016. And field 3 was under that business as usual for at least 10 years.
Well, you know, the good thing about some measurements is they're reasonably permanent. So you could go back and measure that depth to the Claypan.
That is true.
Or somebody else could measure it for you. That would be nice. See if you can convince up and coming undergrad to take that on. So, well, thanks. The part of the presentation I saw was great. So I appreciate you taking the time to give it to the department.
Thank you.
So
I really enjoyed it.
Do we have any other questions? No questions in the chat, but Richard Roth sent a message for you there, Frank, if you didn't see that.
Thank you, and hey y'all.
I guess if there aren't one more call for questions, I guess.
And if there are any other questions, here's my email, or if you see me in the hall, feel free to stop. I'm always willing to chat.
Yeah. So we really appreciate you presenting here today, Frank, and we can look forward to seeing you all here next week at the same time for another presentation. So thank you for coming today.
Thank you. You all take care.